To kick off today's topic, we will have two speakers coming with us. One is Hsin-Fang, another one is Xin-Yi. So today's topic is going to be about how do plants adapt to climate change and pollution. And before we kick off, I would like to speak out some rules happening in this 1 hour of talk. So if you have audience who has any questions, please technical questions, please reach out to IT, which will have see person with IT support on the names. And also you have we will have a Q&A section. If you have any questions, feel free to type in the chat box or save your questions until the Q&A section, and then we will let people to unmute themselves and ask questions. Okay, so today our first speaker is gonna be Hsin-Fang, and she just graduated from agricultural chemistry at the National Taiwan University and just arrived in Boston in late September. And she is now a postdoc fellow in the MIT and is studying effect of climate change on water and then nitrogen use efficiencies of crops. But today she's going to talk us about her work on during PhD period. So let's welcome Hsin-Fang, and it's all yours.
Okay. Hello everyone. I'm Hsin-Fang, and then today I would like to share about Galian Indian Thallium toxicity to plants and environmental risk of their contaminated soil for growing rice and wheat. And so this is my PhD thesis, and then I got my PhD from the National Taiwan University and Academic Seminar. So the Galian Indian Thallium, they are belong to the group 30 in the periodical table, and then among those groups, only the boron is non-metal and metal, and then play an important role in the cell wall. And the aluminum is and none of the group 30 metal qualify as essential to life. Aluminum is the most abundant metal in the Earth's crust, and then while its soluble iron forms show phytotoxicity to plants, so a lot of research is doing on the aluminum toxicity to the plants. And then for the Galian Indian Thallium, they are of interest because of they are widely used in the semiconductor industry. And then for example, like gallium is as a power amplified chip in the cell phone, and just take the iPhone 30 as an example, there's around 30 chips in one iPhone. And for the Indian, it's very special material, which is transparent, but it's have conductive. So every touch screen, they use the Indian as a material. And then for the thallium, is also used in the semiconductor, widely used in semiconductor industry, and also as a isotope tracer, and then used as a medical. There's some medical usage. So because the Taiwan is one of the most important semiconductor industry country, so that's why we are focused on those three metals. And then because the widely used, so when we are processing those semiconductor products, they have there's inevitable that those metals can release to the environment. So indeed, there is a report show that they capture the Formosa squirrel as a bioindicator, and then to measure their Galian Indian Thallium concentration in their tissue in their body, and then the results show that those animals captured in Taiwan, the concentration, those three metals concentration is higher than those captured in Japan. And also, most important is that they find that the animal captured in the Miaoli is higher, show the highest concentrations compared to the other place in Taiwan. And then the author suggest that it's because the contaminants come from the Hsinchu. So as we know that there is a environmental pollution has been reported, so they have an opportunity that they can accumulation in the food, and then so they can cause the health risk to the human. Sorry, and then so there's a knowledge gap, like mathematical understanding of the risk posed by those three metals in the environmental is necessary to protect the human and environmental health. And plants are critical to both ecosystem function and to the human food supply. However, the information on the interaction of the guardian Indian thallium with plants is limited. And so if we want to do health risk assessment, there's two important features we need to take consideration. One is the when those elements are released in the environment, and then what's their exposure pathway, and then when plants face those elements stress, what is the toxicity response? So when those elements are released to the soil, they can participate in the mineral or in the clay, organic matter, or the soil solution, and then their solubility will determine that their availability in the soil, and then their availability is affected by the different soil property, and then including the soil pH, reductive potential, the calcium exchange capacity, or the soil tissue or organic matter concentration in the soil. And then among those soil properties, the pH is play the most important role. Like take Indian as an example, the X is a different pH from the low to high, and then the Y is the Indian's concentration in the Indian concentration, and then the different color, this blue color is mean there is in the soluble phases, and the orange color is mean there is in the solid phases. So we can see that when the pH increase, like the pH 7, the most of Indians is become the Indian hydrous oxide precipitate. So it's like when the soil pH is lower, the metal will have higher solubility, and then they will have higher availability to the plant for plant uptake. And then so speaking of the plant, when plant face those metal trace, because those metal is non-essential for the plants. So if the plant can uptake those non-essential metal, that means those guardian Indian thallium should compete with other essential metals transporter, so they can be uptake by the plants. And then after the accumulation in the roots, they will translocation those metals through the ligand to the shoots, and then accumulation in the shoots. And also those metal is because they are non-essential to plants, so they can cause the oxidative stress, and then make plants show some toxicity phenotype, like the roots will grow inhibited, or they are change the color, like the anthocyanin accumulate, so they can make the plant leaf turn purple. And also there's some strategy for plant to defend those metals. For example, like plant can secrete some root exudation, and then those exudation can prevent plant to uptake those metals. Okay, so based on those background, we have two main questions about the guardian Indian thallium toxicity, and then so there's two questions in my thesis. So one is, how is the guardian Indian thallium toxic for the plants, and then what is their bioavailability in soil, their accumulation in the crop, and then the health risk imposed. So for the first question, we use the model plant called Arabidopsis thaliana, and then we we grow in the normal condition for 7 days and then treat for the Ghanaian, Indian or Thailand for another 7-day treatment. And then we will do some physiological and molecular response analysis. And then for the second question, we select 3 different soil, and then those 3 different soil have different pH. Like we choose because as I mentioned, the pH, we think the pH is the most important role to determine their solubility in the soil, in the environment. So we choose the 2 acid soil is called the Pingzhen and Tainan. Like we can see the pH is very low in these 2 soil. And then one alkaline soil is called Taikang, and then the Taikang's pH is quite high compared to these 2. And then for other soil property like the texture or organic matter, all this this is mean the critical ions in the soil, and then this is non-critical. And then for other soil property, they are very similar, but it's not the same. But so for this for this experiment, we have a hypothesis that the Ghanaian, Indian, Thailand in the PC and Taikang soil will show the higher bioavailability, and however for the Taikang will show low bioavailability. Okay, so here's some results. So the first, the take-home message is that the effects of the Ghanaian exposure in epidaxis is similar to the aluminum. This because we find that when the epidaxis and then they face the Ghanaian stress, they will secrete the C3 and methylates, those 2 exudation, and then the C3 and methylates, and then for this behavior is quite similar to when plants face to the aluminum toxicity. And then the underlying mechanism is that the C3 and methyl can complex with the Ghanaian, and then the Ghanaian and C3 complex is too big, the plant cannot uptake. So this is the main. So this is the observation that the plants tolerance to the Ghanaian, and then we find that after the plant uptake the Ghanaian can accumulate in the intercellular space, and then like this data show, and then we can see that this is a root tissue, and then this spots, this is a Ghanaian accumulation in the intercellular space, and then after we see this signal, and then we do the index analysis to see what's element composition in those spots, and then we find that there's the red spectrum. This is a Ghanaian's intensity, so we know that this is a Ghanaian. There's a Ghanaian accumulation in the intercellular space. However, if we check the cytosol part here, and then there is no Ghanaian signal, so we know that so this result can confirm that the Ghanaian only accumulates in the intercellular space. And then this is another strategy for plant, is because if they can accumulate and precipitate in the intercellular space, they can reduce their bioavailability to the plants, so can reduce their toxicity to the plants. Okay, and then so for the Indian, we find that it is similar to the Ghanaian, because the Ghanaian, Indian, aluminum, they belong to the group 30 in the periodic table, so they share similar chemistry properties. So so for the Indian, the plants have a similar tolerance strategy, like the plant can secrete the similar oxidations, which can prevent the Indian uptake. And then after the Indian uptake accumulate in the roots, we find that they share a similar pattern of the Ghanaian. They can accumulate in the intercellular part in the root tissue, and then this red signal is all the signal come from the Indian. And then also we find that the Indian can translocation to the shoots, so the ligand, and then accumulation in the and then when they translocation to the leaf, they can accumulate in the major sites of the leaf tissue. And then the most interesting thing is that we find that under Indian toxicity, the plants leaf turn purple, and then we find that it's accumulation of the anthocyanin. The most interesting is because this anthocyanin accumulation is very similar when plants face the phosphate deficiency. So the next question is that this anthocyanin accumulation is caused by the Indian toxicity or caused by the phosphate deficiency. And then the results show that we find after we analyze the phosphate concentration in the leaf tissue, we find that under Indian treatment, the phosphate concentration is quite lower compared to the control plants. And then we find that this is because the Indian can take one of the root to show translocation, phosphate transporter called the 4-1. So when under the Indian toxicity, the Indian can attack this transporter and then make these transporters show lower stability, and then so that's why they can under Indian toxicity the lower phosphate can translocation from the root to shoot, and then so under Indian treatment, so the anthocyanin accumulation is caused by the phosphate deficiency, and then phosphate deficiency is caused by the Indian toxicity. Okay, and then so the thallium, and so as for the thallium, for this part of experiments, we do the analysis for the thallium exposure to the thallium exposure plants, and then we find that the under the thallium toxicity, the plant, the thallium can cause the shoots turn yellow, and then we find that this is because the thallium will affecting the this light harvesting complex 2 in the chloroplast. So that's why the thallium so the thallium can make plants leaf turn yellow, and also we find that the thallium will cause just oxidation, and then they have anti-oxidants called the glutathione, and then the glutathione can reduce those oxidative stress, and then because and then we find that because the thallium is uptake through the potassium transporter into the plant tissue, and then this is because the thallium and then potassium share the similar properties. So this is a key result for the thallium's toxicity to the plants. Okay, so we use those model plant, and then we show that the Galian Indian thallium they do cause the toxicity to the plant tissue to the plants. So next we want to note that in the the true experiment, the true conditions, what is those metal will cause the toxicity to the crops. So as I mentioned, we select 3 different soil to do the experiments, and then because of the Galian Indian thallium, they are sure very similar results. So here I just show the Indians result in these soil experiments, and then we find that the Galian Indian thallium, when those Galian Indian thallium released to the soil, most of those metal is they will participate and then in the soil, and then no matter what the pH is. So this is a little different from our first hypothesis that the pH is play the most important role, and then because our results show that no matter what pH is, they all show the low bioavailability. Okay, but however, we find that after the rice and then with uptake those metal, they can translocation and accumulation in the grains, and then most importantly, they can the metals is accumulate in the endosperm. So those endosperm is the edible parts of because we eat here. So if the Guardian Indian and Thalian is accumulates in the endosperm, that means the consumption of the Indian Guardian and Thalian contaminates grains, thus concern for the human health. Okay, so for my thesis, I think the main contribution is that we systematic and mathematically investigate the risk associated with the Guardian Indian Thalian, those emergency and prevent contaminates. And we provide important data for police making about the Guardian Indian Thalian soil contaminates and the overall environmental risk that society face. And we also provide a framework for the public in recognition and understanding environmental risk and when to take action to ensure the human and environmental health. Okay, and then so now I am a postdoc as a CE at MIT, and then this is my professor David Desmarais. Okay, so our lab is focused on the food and water security. And then for my project, I'm doing my project topic is understanding the role of the climate stress on the plant metabolic pathway. And then we are focusing on the water and nitrogen use efficiency. And then the start point is that we can show this two figure. This is the edible part, the nutrients concentration in the edible tissue. And then this is in the leaf tissue or in the wheat. And then and then the S is the change in the concentration. Change in the concentration of those nutrients in those tissue, and then they compare to the high CO2 and then the nowadays the high CO2 and nowadays CO2 condition, and then they find that the high CO2 can decrease all the nutrients elements in the plant tissue, and then the most significant is come from it's nitrogen. And then it's very interesting is because as we know that under the high CO2 condition, the plant can have high photosynthesis, so which can increase the biomass of those tissue. So one of the hypotheses of those phenomena is that the dilute effect. So that is because when the plant biomass is increased, those nutrients will be decreased. But if this is the reason. If this is the reason, that means all the nutrients should decrease in the same degree. However, we can let the nitrogen decrease have a most significant decrease. So that's why we want to make sure in the future climate change stress. How can we maintain the nutrients quality for our food to ensure the food security? Okay, thank you.
So thank you, Hsin-Fang, for sharing such a wonderful talk. So we can go back to gallery mode, or any audience, any questions can unmute themselves and ask question now. Okay, well, I see one raise hands. Please go ahead. Xin-Yun.
Hi. Hi, Hsin-Fang. I have a question. I think maybe you mentioned, but I just missed out. I know a lot of times that when the plant roots absorb or take in the ions, they also work with some bacteria and other organisms. Do you know how those pollution will affect those bacteria that work with the plants?
We don't know because we don't do any bacteria in the rhizosphere. But I think it's interesting is that because under those stress, they can induce the roots to produce some more exudation, like the citric metals. So and then those citric metals can become one of the carbon source for those bacteria. So maybe I guess it should change, but we don't know how those change.
Thank you.
Thank you for the questions. Anyone else? So if not, I will take over the time. So can we start the sharing? Before we jump into the second speaker, I would like to invite everyone online now. We are going to have a photo time. If you will ready to turn on your camera and join us for our photo shooting, and please feel free to do so. And then we will have our IT support to take a photo for us. Okay. Let's count down. 3, 2, 1. 3, 2, 1. Okay. Thank you. Do we get it? IT support. Good. Okay. Let's move on to the next speaker. Our second speaker is our speaker is Xin-Yi, and she just also doing a postdoc here at Harvard Medical School, and she attend a school in Taiwan to get her Ph.D. from the Taiwan International Graduate Program at Academic Seminar. And she's now working as a postdoc at ISA in Harvard and studied the plant's nutrition and the energy signaling via Tor regulatory network. So let's, to a short of time, let's just move on. She needs to take over.
Thank you for the kind introduction. I'm Xin-Yi, happy to be here to share you my recent publication, which is entitled "To Prepare for the Hypatia." Ethylene, which is the plant phytohormone, control the translations through 2 key factors, GCN2 and EN2. Oh, just give me a second. Control the 2 key factors, GCN2 and EN2, under the submergence. Okay, so before my presentation, to introduce my story, I want to use this cover to remind you like what happened in this year in July. There's a big storms threaten the South United States, and also in the East Asia also encounter these kind of big storms. And okay. So as you know, like during the typhoon and hurricane seasons, there was a big flooding caused by this heavy raining in the south part of the United States, and also in East Asia, like Pakistan, India, and Taiwan. Okay. But the frequency of this kind of flooding happened more frequently now because of the global warming and climate change. Okay, so like this year in the Pakistan, almost as I remember, almost 90% of the Pakistan submerged by the water for a month. So you can imagine that the pub also caused a lot of big loss in the agricultures. I also read the news about the Indians. The Indian farmer even commit suicide after these big floodings because they cannot afford the loss of the crop productions. Okay. And here also I have one statistics data which studied in the United States during these 10 years. You also can see that the cost loss of the flooding increased 2-fold in these 10 years. And you also can imagine that we also have a kind of big issues in Taiwan. So how could we secure the food product in this kind of extreme weather beyond stop the extreme weather coming, or like the reduce the carbon emissions? We actually can do more. We can try to develop the new plant breeding structures, like to create or select the more tolerant crops to adapt this kind of extreme weathers. So to get this kind of stronger crops, we need to understand how plants adapt these stress conditions. Okay. So my study mainly focus on the plants under these flooding or submergence conditions. Under these submergence conditions, the pathogens in the root in the plant tissues would decrease within the hours. Okay. And actually, plant and animal all have a quite conserved oxygen sensing mechanisms. Like the animal, they will use the Ph.D protein, and the plant use the PCO protein to sense the oxygen level change to determine the master regulators called HIF1 alpha in mammalian and or the EF7 transcription factor in plants. And these two master regulators, the stability of these two master regulators determine the expression of the hypoxia response genes. Okay. On the other hand, because of limitations of the oxygens, the ETC, the electron transport chains in the mitochondria also be shut down. So that means that the energy production also be affected. Then the cell will change to use the anaerobic metabolism, which means they use the fermentation used to produce the ethanol and then get ATP. Okay, and furthermore, cell will also shut down the translation to conserve the energy usage. Okay. So still in the animals, because there's a lot of this kind of stress also correlate with the cancer study. So they already categorized there's one key kinase called AMPK. It can sense the energy status change in the cell and then further shut down the toll signaling and recruit the 4EBP, this key protein, to disrupt the translation initiation complex formations. However, we couldn't find these kind of proteins in plants. So how plant cell repress the translation during the hypoxia is not clear for the decades. So in the beginning, we use the acidosis as our study material. This is a kind of a small wheat plant, and the cell cycle is quite short, so it's good for the molecular study. So we generate this AMPK ortholog gene mutants in acidosis and perform the phosphoproteomics to compare and compare the mutant and wild type profile to identify the possible targets of this kinase in plants. And luckily, we got about 1,800 unique phosphopeptides, and that also include about 50 proteins show different phosphorylation patterns in the wild type and mutants. Okay. And they're most then involved in the metabolism process and also the stress response. And one of them catch our eyes is the protein involved in the protein synthesis. Okay. So we do further study and find out that the SNRK1 can phosphorate the plant-specific translation initiation factor called EIF iso4G1, and to enhance the specific RNA translation, like the two key enzymes in the fermentation pathway, the ADH1 and PDC1, that to ensure the protein production to adapt the stress. Okay. So till now, we know in plant, they have a mechanism to enhance the specific RNA translations. But we still don't get the answer like how plant cells shut down the translations. Okay, so luckily, we all, as a PhD student, we always have the plan B, the second projects. So I parallelly work on the other kinase called GCN2, which also quite well characterized in the yeast and the animal systems. It will be activated during the kind of nutrient starvation or hypoxia condition, and phosphorate one of the subunit of the EIF2, which is the in translation initiation factor 2 alpha subunit, and then stop the cycle of the GTP cycle, then reduce the translation initiation complex formation to screen, and then further stop the translations. Okay. So to our surprise, didn't expect that the EF2 alpha phosphorylations activate within the 15 minutes of the submergence. Okay. And it could be until 2 or 3 hours of submergence and then further go down after the submergence conditions. And we also check whether these 2 alpha phosphorylation indeed controlled by the GCN2 through the genetics tool. We check the phosphorylation in the GCN2 mutant and the 2 overexpression GCN2 light. And you can see that the phosphorylation disappears in the GCN2 mutant background. Okay. So we also need to also perform the sucrose gradient fraction to celebrate the ribosome and the polysomes. So in this region, the big picker, the big picker indicates that more RNA now is under translation process. Okay, so you can see here, this is the wild type plant. Under the submergence, the translation really show reductions. Okay, but in the GCN2 mutants, the reduction is minor than the wild type under the submergence, and its pattern can be overcome by overexpression of GCN2 proteins. You can see these 2 overexpression lines. They show clearly a translation reduction. Okay. So from here, we need support this data support that the GCN2 regulates the translations under the submergence. Okay, and so then we further examine whether this is kind of regulation indeed critical for the tolerance of submergence. We quantified the 2 energy metabolites, ATP and AMP in the mutant and overexpression line. You can see here in the overexpression line, they actually contain higher ATP amounts during the whole stress conditions first durations. And in the mutants, the ATP decreased after the 6 hours of submergence. And also the mutant accumulate more AMP. This indicates that in the mutants, they lost the control to balance the energy. They cannot conserve the energy usage under the stress conditions. Okay, so we also perform the survival rate test, and we check the 2 stages of these plants. One is in the seedling, and the other is in the mature plant stage. And you can see the pattern is quite also consistent with what we observed in the previous data, like the GCN2 mutant lost the translation repressions, show more sensitive to the submergence. And overexpression line can rescue this phenotype. And in the mature plant stage, also show a similar pattern. And we also test 2-day, which is a longer submergence stress conditions. You can see that almost all the wild type died after this 48 hours of submergence in here, this page, or almost half hour. Here still have a few of us wild types still survive, but compared with the wild type, the overexpression line still can maintain the green leaf and can recover after the 2 days of submergence. So here we support and prove that the GCN2 is critical for the submergence tolerance. Okay, so the following we keep search that. So what kind of signaling factor control this GCN2 signal under submergence? We actually do a lot of screening and find this ethylene could be our potential candidate. Okay, ethylene, as I have told you in the beginning, is a gas hormone. It's a plant critical fat hormone. It's a gas form hormones. And here's a fun fact. When you burn the incense stick in the temple, actually you already smell a lot of the ethylenes. Okay. And this gas hormone controls a lot of control. It's critical and control the plant growth, development, and stress response through the whole plant life cycles, including like the fruit ripening and flower senescence, and also the flooding stress tolerance. Okay. In the plant tissue, this ethylene synthesis from the mesalins and further they will be released and diffused out of the cells. Okay, and in the previous study, they already observed that under the submergence, in the early stage of submergence, the ethylene is trapped in the submergence tissue because it's a gas. It's difficult to diffuse out of the cell to the waters. Okay, and in 2019, it even be proved that can control the stability of the master regulator under the hypoxia conditions. Okay, so we test whether 2-alpha phosphorylation can be activated by the pure ethylene treatment. The answer is yes. You can see here 30 minutes of treatment already activate the 2-alpha phosphorylation. And then we also treat the plant with the ethylene receptor inhibitor to block the sensing of the cells, ethylene sensing of cell, and then treat the plant with the submergence. And so here you can see that the phosphorylation show reductions in the inhibitor treatment samples. Then we also use the genetic tool to test whether indeed the ethylene control the EF2-alpha phosphorylation. We use the 2 receptor mutant and the data to examine whether the 2-alpha phosphorylation indeed disrupt, and the answer is yes. But here but further, we also test the other 2 the downstream factor mutants of these ethylene receptors, that EN2 and EN3. But you can see here the phosphorylation didn't show any different patterns like the wild type. So we are kind of scary at that time, So we further examine whether the translation repression show different patterns in these mutants, and more weird results showing here that we find that indeed in the ethylene receptor mutant, the translation reduction abolished, but it also abolished in the EN2 mutants, and so we further generate the EN2 and GCN2 double mutant, and you can expect that the translation reduction totally disappear. Okay, and we also examine the 2-alpha phosphorylation in double mutant. Okay, so definitely there's no 2-alpha phosphorylation here. Okay, and so we also perform another strategy to examine what we observe is correct or not. We use the click technique to label the newly synthesized proteins, and in this wild type and single mutant, double mutant. You can see here after the hypoxia treatment, the newly synthesized protein really reduced in the wild type, but it still shows the reductions in the single mutants, but the reduction is minor, even no change in the double mutant. Okay, and the phenotype also indicates that indeed the single mutant already shows certain level of sensitive to submergence compared to a wild type, but the double mutant and the receptor mutant show the stronger damages than the single mutants. So this tell us that probably the EN2 and GCN2 are critical under submergence condition to regulate the translations. Okay. So we perform the transcriptome and translatome analysis. Here is the translatome we select the whole set of gene which show the translation repressions under the submergence. And you can see that basically the EN2 in the EN2 and GCN2, you already see there's no differences. There's no repressions like the GCN2 double mutant. Okay, and these genes are basically all involved in the translation and the growth relative response, which support that indeed under the stress condition, plant will start to grow and conserve the energies. Okay, and And this RNA also share the similar features in their 5' and 3' UTR regions. Okay. We also examine, we also check the other set of gene RNA which show the translation enhancement under the submergence. And here we also didn't see the different patterns in the single mutant. Basically, the data tell us that the EN2 and GCN2 both can control these genes translations. Okay. Then when we further go to upper stage to check the transcriptome analysis, here we finally find something interesting. You can compare to the whole profile and we find that the GCN2 and EN2 can regulate different things. Like in the GCN2, it major control the transport cell elongations process. And in EN2, it will shut down the cell cycle and also trigger the ethylene and the hypoxia gene expressions. And there's one more thing I want to mention here, that if we also dig out the 15 minutes of transcriptome and find out that within the 15 minutes, already about 1,000 genes have been activated through the EN2, and also a subset of gene also shut down by the GCN2. Okay, so and we also check about these genes geotin that you can see here. Within the 15 minutes already, that of the stress, especially for the hypoxia responding gene, already activate. Okay, so I hope you still remember I mentioned in my introduction that plant enter the hypoxias. It takes about 1 hour, then plant will enter the hypoxia under the submergence condition, because it still can use the oxygen which generated by the photosynthesis. So why plant turn on the hypoxia within the 15 minutes under the submergence? Hypoxia response within the 15 minutes of submergence. So we come out with a model, and this model also be admitted by ENVO, and they write this model in their news. So I just borrow their picture to share you as a take-home message on my whole story. So first, during the several generation, plant learn lessons from the nature that when the big rain comes and cause the flooding, submerge the tissue, and restrict the gas like the ethylene diffuse out of the cell. This kind of trap ethylene tell them that awake them that, oh, there will be a kind of more severe or different stress comes, and this would be the kind of hypoxic stress. So they further prepare for the coming stress and turn on the two pathway. This is through EN2 and GCN2, and GCN2 can phosphorate the initiation translation factor 2 alpha and to stop the translation complex to screen and select the specific RNA. And further, there will then the EN2 further will also participate in the general translation repression and activate the stress response gene to adapt these hypoxia conditions. Yeah. Okay. So thank you. So this is all my story. And here I would like to thank my grant funding for the whole projects, and also all the core facilities support for this story. And especially I would like to thank my other PhD advisor, Dr. Shi, and my co-first author, Mei Yi. She helped me to maintain and take care of the plant material in the GCN2 projects. And here I also want to especially thank my current PI, Shen. She allowed me to finish the whole story when I'm here. So, and then at the end, thanks everyone to pay attention, and I'm welcome to take any questions. Yeah.
Thank Xinyi again for such a nice talk. So we'll take any question from the audience now. Please raise your hand or, okay, we got one question from Ginny. Please unmute yourself.
Hello.
Hi.
Yeah.
Yeah.
So thanks for your beautiful talk. I have two questions here. One is about the result of the Western blot after the treatment from of the ethylene. So I see the expression from, I think that is from the your photoreceptor promoter. Could you pull up the?
Let me share my screen again. Just give me a second.
Thank you.
Okay. Yeah. So which?
I noticed that when it is
Mhm.
after 30 minutes of your treatment, you can see a big expression, but I don't know why, like when time prolonged to 60 minutes from your Western blot, you really couldn't see the band.
You mean the 2-alpha phosphorylation or? Actually it's a time course. We check 30 minutes, 1 hour, and 3 hours, and then we stop the treatment and take the plant out of the water. So you will see the phosphorylation increase to 3 hours of treatment, and then decrease when we take the plant out of the water for 30 minutes. So you will see that kind of increase and decrease.
Oh, I see.
Yeah.
Okay. So my second question is that so now we know the mechanism and how we can like enhance the ability to enhance the plant's ability to resist the submersion, but how can we apply to to reality? Like how can we do to improve their ability in real world?
This is a great question. So, I have to say, like, if the society can accept the GMO, then we do have a lot of candidate, like we can improve the plant resistance and to like the gene editing certain crops, and then making it more resist to this kind of flooding stress condition. Because how the reality is that the people still resist the GMO plant crops so far. So on the other hand, now as we have learned from plant that the plant will use the ethylene as a signal to enhance the gene response. So we have an idea. And we also have been proved by other groups that we try to pre-treat the plant with the ethylene. That means we activate the stress response in advance. And the other group already published this result that indeed they can enhance the plant to tolerance this submergence and can enhance about 50% the survival rate. So that would be another way to save the crop productions under this stress
condition. Interesting because I think if we produce like pretty much of the ethylene, it might save the plants, but it might cause global warming, right? Cycle.
That would be enter this kind of cycle, right? So another way is that that's a great question. So far I didn't have a good answer for that. But you have to find a balance, to save the world or save the people.
So there's any questions? Okay.
I want to ask a question following Jenny's questions. So if you pre-treat with like ethylene, is there any like downside? Would that hurt the plant or like any disadvantage of that?
Pre-treat the ethylene. Let me think. So far, what we have no that pre-treatment. There's no clearly downside. But let me think. But I have to say that the duration of the pre-treatment is quite critical. If the plant retain high ethylene concentration for too long, the growth of the plant will be will retard. Yeah, so the pre-treatment duration is still also critical. Yeah, so maybe this is the downside of the pre-treatment. So we need to adjust the best duration, concentration of the pre-treatment to enhance the tolerance, but not cause other growth retardation effects. Yeah.
Thank you.
Okay, thank you for all these discussions. These are all good questions. So it's due to the limit of time, so I think we almost come to the end. So before we go away, I would like to again do some promotion. If you can look into the meeting chat box, we do have some upcoming activities, especially one that's career development series will happening in December 3rd, Saturday. And please stay tuned with us, either come in person or join us online. And also don't forget to follow us on Facebook. We do have a website and also LinkedIn. And finally, thank you for everyone to join us for tonight. All right. Thank you all. Bye.